Next up, we have Dr. Louise Cameron, who graduated from Northeastern last month, and she'll be talking about her work on marine bivalves and their response to ocean acidification.
Okay, so yeah, my name's Louise Cameron. I just finished my PhD at Northeastern, same as Jess. You can find me on Twitter at LouiseCam93. So I'm really interested in understanding how scallops can cope with climate change. Now, you probably all recognize scallops as these delicious morsels that you'll see on your plate. Scallops are a super important species for US fisheries, but they actually normally look more like this. So a scallop is a bivalve mollusk. Bivalve means that they have these two shells. I think that they're a really charismatic and interesting bivalve. They actually have over 100 eyes. If you notice those little black dots along the edge of their shell, those are their eyes. They can also swim by opening and shutting their shell really fast. So they're a pretty fun animal to study. As I mentioned, they have these two large shells. These shells are made of calcium carbonate. And because of these shells, this species is particularly vulnerable to the effects of ocean acidification. They build these shells from calcium and carbonate ions in seawater and combine them to form calcium carbonate, which is a mineral. However, ocean acidification is limiting the amount of biologically available carbonate in the water column, and without this carbonate, they can't build their shells as well. So as I mentioned, this species is really important for our economy. In 2018, their fishery generated more than $400 million in profits. And a lot of that revenue is generated right here in Massachusetts, where I'm based. So it's really important for our local economy. The Atlantic sea scallop fishery takes place up and down the east coast of the United States, but a large portion of it takes place on George's Bank, which is this elevated region that extends around about 200 kilometers offshore. You can see we've got Massachusetts in the top left corner up here with Cape Cod sticking out. And George's Bank is a really interesting region because it has very complex oceanography. So it has this strong depth gradient from the shallowest depth being around 30 meters, ranging down to 200 meters where it drops off the continental shelf. It has strong tidal currents. It's subject to upwelling of low pH deep water onto the bank. And it has a wide variety of sediment types, ranging from sand, shell, and gravel that can buffer local conditions at the benthos. So because this region has such complicated oceanography, it's likely that it also has variations in carbonate chemistry across the bank. So variations in the amount of that carbonate ion that these bivalves use to build their shells. However, George's Bank is a bit of a data gap. We have a lot of pre-existing data on carbonate chemistry in the Gulf of Maine to the north and in the mid-Atlantic bight to the south, but not as much on George's Bank. So for my research, I first wanted to quantify the state of things on George's Bank. So I went out on a commercial fishing boat and I collected seawater samples at 15 sites on the bank. I collected them from depth and then I measured the carbonate chemistry of the seawater. So how much of that carbonate ion is available for the scallops. At each of my sites, I also collected 50 scallops, and then I looked at different properties. So I looked at their shell thickness, shell strength, tissue condition, gonadosematic index, which is a measure of reproductive output, and I also measured their growth rate by counting age lines on their shells. So this allowed me to survey the current state of Atlantic sea scallops across their range on George's Bank. I'll show you a couple images, a couple figures from that in a moment. The other side of my PhD was doing a tank experiment. In the marine lab that I worked at, we have this really cool ocean acidification array. In each of these tanks, we can bubble mixtures of carbon dioxide and air into the seawater at different concentrations. And this allows us to simulate future conditions. So we can take a look into the future and put different marine organisms under these scenarios. We can also alter temperature as well. So in my experiment, I kept scallops at 3 different temperatures and 3 different CO2 concentrations. that simulated present day, near-term future scenario, and an extreme acidification scenario. And again, I didn't put much data on my slides, but in this experiment, I found that their calcification rates declined under ocean acidification, so they weren't growing as fast. In fact, in my most extreme acidification conditions, they were actually losing shell weight. So their shells were dissolving back into the seawater. I also found increases in mortality, both in response to temperature and CO2. So warming and ocean acidification together were causing increases in death rates amongst my scallops. Now I'm just going to show you one of the starkest results that I found in my field studies. So I also had access to a scanning electron microscope that allows me to take really high resolution up close images of the shell's surfaces. So this is the inside of a scallop shell at 15,000 times magnification, and each of these little rectangular things is a calcite crystal. Calcite is the type of calcium carbonate that scallops use to build their shells. So you can see in this image, these crystals are really well defined. They have nice uniform edges. They're overlapping a lot. And this gives the scallop shell a lot of structural integrity and makes it really strong. This image is taken from our highest carbonate saturation state site. And now I'm going to show you an image from our lowest saturation state site. And you can see in this image that those crystals are really disorganized now. It's hard to tell one crystal apart from the other. They have signs of dissolution along the edges. I also tested the strength of these shells with an Instron, which is a type of crushing machine. And I found that the shells from our lower carbonate saturation state site were significantly weaker than at our higher carbonate saturation state site. So this means that these scallops could be easier targets for predators in the future. Yeah, that's all I've got, and I'll happily take questions in our discussion. Thanks.
So we'll jump on over to Dr. Jessica Tarosian, who graduated from Northeastern in April and studied blue mussel responses to climate change and is now a policy analyst at DOT.
Good morning or good afternoon. Thanks everyone for coming today. I just completed my PhD in the Helmuth Lab at Northeastern University at the Marine Science Center in Nahant. And my research focused on blue mussels, as Helly mentioned, but sort of within the framework that we do in that lab of ecological forecasting. So let me just tell you a little bit about ecological forecasting. And so basically the idea behind ecological forecasting is that we're using a combination of field experiments, lab experiments, and some computer models to try to understand how species are going to respond to future changes. And similar to what Stephen was talking about, we basically in the lab can really stress out the animals and do a stress test similar to what your doctor would do for you at a hospital. We test heart rate, we test feeding rate, respiration rate, and then also sort of how well an organism is able to assimilate the energy that they're taking in from food. And then we can use these in different models, such as dynamic energy budget models or scope for growth, to try to understand how species are going to grow and reproduce under these conditions that we're seeing in the field and in the lab. One of the things that's really important is that we're looking at, when we're looking at these animals in the intertidal, we are trying to understand kind of what their thermal response is. And unlike us, we regulate our body temperatures. Most of these species are actually subject to weather conditions. They're what we call ectotherms. And they rely on their shape and their size to sort of modulate how they respond to things like air temperature, solar radiation, wind speed. And you can see from this presentation that the way that two different animals on the rock at the same spot might be experiencing very different temperatures. For example, a mussel might be much, much hotter than their predator, a sea star. And I'm actually realizing I'm sharing the wrong presentation with you. So I'm going to very quickly stop sharing. And then I'm going to jump back into the presentation that I intended to share. Sorry about that. My apologies. There we go. That one was going to be much longer. And so I'm going to jump ahead. So to try to correct for these issues with thermal temperatures that we're reading in the field, we create what we call robologgers. And what we're trying to do is match sort of the thermal inertia and the thermal conditions of a temperature logger to the actual biological way that these animals are experiencing temperatures. And unmatched loggers, so you can see that this logger in the lower left-hand corner is small and shiny, and that's going to interact with those environmental variables in a very different way from a mussel, which is, you know, a little bit bigger, black, in absorbing solar radiation in a very different way. And you can actually get unmatched errors of, you know, greater than 14 degrees Celsius. But when you start to try to mirror those thermal conditions, you actually get much closer to what the animal is actually experiencing in the field. And in this case, what we're trying to do is match the conditions of the blue mussel. And similar to what Stephen was talking about, blue mussels are really important from a fisheries perspective. They're very important to the Gulf of Maine shell fishery, as well as Canada. They're also what we call a cosmopolitan species. They're distributed all over the world and are very important, not only for food for humans, but for other species as well. They're foundational species. They help bring in nutrients from the bottom up into an ecosystem, and they form these really dense bed habitats. They're also a really good sentinel species for climate change because they have such a broad thermal tolerance range that you can see here.
Hey Jess, we can't actually see your screen anymore.
Oh, I'm so sorry about that.
That's okay. I didn't want to interrupt.
Thank you. As we were just talking about problems with technology. Let's see, that's better. There you go. So yeah, the thermal tolerance limit is, you know, and I've actually measured thermal tolerance actually higher and lower than this. It really depends on sort of how long these animals are exposed. But the mussels in my experiments are dying in air at around 42 degrees C and are surviving, depending on the population, down to even -17 or -19 C in the winter. So during my experiments, I wanted to look at how mussels in the Gulf of Maine were doing. If you talk to a lot of researchers in this area over the past 30 years, they've noticed a lot of declines in mussel populations. So I split up the Gulf of Maine into two regions, the north and the south. And typically we would expect to see a thermal gradient in the temperatures that these animals are experiencing. But my professor did work on the West Coast and found that mussels are actually, because of the inconsistencies in tidal timing, as well as sort of how they're shaped and how they're oriented on the rock face, there's actually a lot of patchiness and there's some unexpected hotspots. So this is what one of these looks like in the summer in northern Maine. They're very dense. They're very different than what we see sort of down here closer to Massachusetts, but they're still thriving kind of up embayments. And I wanted to understand why this was happening. So I deployed a bunch of those temperature loggers that I think you weren't able to see, but this is what they look like. I ended up making my own system to be able to get 76 loggers out in the field in a variety of conditions as well. We wanted to try to understand how these animals were surviving, not just in the summer to extreme heat stress, but in the winter to extreme cold stress. And what I found is that in the summertime, we're typically seeing that gradient pattern as you would expect. But in the winter, during really severe cold snaps like this one that you're seeing depicted in this picture, this ice in the north actually acts as almost an insulator. And so counterintuitively, in the winter, during really severe cold snaps that are likely to result in high mortality, the mussels in the north are more protected and tend to stay closer to 0 C. And in the south, because this ice barrier isn't as... So that's just part of the research that I did. I didn't really include much results because I felt like, having just offended, I could have talked for a very long time. But I did also want to say, since we did shut down last week, just would really encourage you to go to shutdownstem.com and check out their resources for improving Black lives in STEM, as well as trying to help end systemic racism in academics.
Thanks for watching. For more information, visit our website at www.futurefrogmen.org.